During this video, we will discuss how DNA can be analyzed and used in forensic science. The process is called DNA fingerprinting or DNA profiling. Since each individual has a unique sequence of DNA, a person's DNA profile is considered individual evidence. DNA is biological evidence that can be analyzed from person to person for the presence of specific traits. The technique developed by Alec Jeffreys in 1984 was intended to help resolve paternity in immigration cases. However, DNA fingerprinting has a wide array of uses beyond forensic science, including the study and diagnosis of inherited diseases and population genetics. There are actually two types of DNA fingerprints. For right now, we are going to focus on variable numbers of tandem repeats, or VNTR, which is the older method of creating a DNA fingerprint. We will discuss the more current method of DNA analysis, short tandem repeats, or STR, in more detail at a later time. Remember that DNA is composed of a specific order of the nitrogen bases: adenine, thymine, guanine, and cytosine. Some sections of DNA carry the instructions for making a protein, but most of a DNA molecule does not code for anything. In these nonsense sequences, the nitrogen bases often form repeating patterns. So every 9 to 20 bases, the pattern would repeat. Alec Jeffreys called the repeating sections of DNA VNTRs and found that these sections of DNA were unique from one person to the next. Both VNTR and STR DNA profiles can be used to match tissues and match inherited characteristics. Here are some examples of the uses of DNA fingerprinting. This is a DNA fingerprint showing the DNA from a mother, a child, and two possible fathers. The actual father can be identified by comparing the profiles. More on that in a second. This is a DNA profile from tissue samples, like a blood sample, from a crime scene, a victim, and two possible suspects. The DNA profile of suspect 1 matches the DNA profile of the tissue sample found at the crime scene. Since a DNA fingerprint is individual evidence, this profile would place suspect number 1 at the scene of the crime. Now, that does not mean he necessarily committed the crime. Okay, this is all great, but how is a DNA profile created and analyzed? Well, first you need a sample of DNA. You can get DNA from most bodily fluids, including saliva, urine, blood, and semen. DNA can also be found in skin cells and hair follicles. Oftentimes, only a trace of this biological evidence will be found at a crime scene. Thankfully, a biotechnology technique known as the polymerase chain reaction, or PCR, can be used to take a small amount of DNA and create millions of copies that can be used for testing. We will save a thorough discussion of this process for another video. So we have collected some biological evidence at a crime scene. How do we create a DNA fingerprint? The first step is to extract the DNA from the cells. Then, if necessary, you create millions of copies of the DNA using PCR. After that, you create the profile by performing gel electrophoresis. Finally, you make the profile visible by treating the DNA with probes or a dye. Extracting DNA is actually pretty simple. The DNA just has to be separated from other cellular components like lipids and proteins. The white globs in these photos are DNA that has been extracted from peas and tomatoes. A similar process can be used to extract DNA from human tissue or any cell that has a nucleus. The second step is only necessary if a small amount of biological evidence was recovered from the crime scene. As I mentioned earlier, PCR is a method of copying DNA and making millions of identical DNA molecules that can be used for a DNA profile. Once the DNA is extracted from the cells and there are enough copies of the DNA to work with, a forensic scientist can start the process of creating a gel. The first thing that needs to be done is the DNA must be treated with a restriction enzyme. Restriction enzymes cut DNA molecules at specific DNA sequences. The restriction enzyme EcoR1 recognizes the sequence CTTAAG and cuts between the A and the G. Treatment with a restriction enzyme ends up creating DNA fragments of different lengths or weight. Then these DNA fragments of different size are loaded into the wells of an agarose gel, which is kind of like Jell-O, only agarose comes from seaweed. The gel acts like a sieve, like when you want to strain the pasta you just cooked. Small DNA molecules are like water. They move easily through the gel like water moves easily through the strainer. Large DNA molecules are like the pasta. They get caught up in the gel and they don't move as far. Now, gravity is used to separate the water from the pasta, but to separate the different sized DNA molecules, electricity is used. That is why this process is called gel electrophoresis. When DNA molecules are dissolved in water, they actually have a negative charge. When a current is applied to the gel, the DNA molecules are attracted to the positive terminal, which is called the anode. Remember, the smaller DNA molecules will move faster through the gel, so the DNA ends up separating based on the size of the DNA fragments. Shorter DNA fragments will be towards the bottom of the gel, while larger DNA fragments are toward the top of the gel, closer to the wells. Now, in order to see the DNA fragments, you have to complete the final step of a DNA profile. To make the DNA fragments visible, the DNA in the gel must be treated. This could be done with a dye that adheres to the DNA molecule, making it visible, or with probes that contain radioactive phosphorus atoms. The radioactivity makes the DNA fragments visible when ultraviolet light is used to visualize the gel. Anyway, after treating the DNA fragments, a DNA profile is created. Now let me explain this gel to you. The first column of black bands represents the DNA found at a crime scene. The next 4 columns of black bands are DNA from each of 4 different suspects. All the DNA samples were treated with the same restriction enzyme and ran through the process of gel electrophoresis. The DNA fragments were stained with some type of probe, and now the DNA fragments are visible as these black bands. This black band here is made up of thousands of DNA fragments of the same length. Since these DNA fragments are all of similar size, they moved through the gel at the same rate and formed a band of DNA. The different bands in each column represent a DNA profile. This is the DNA profile for suspect number 1. This is suspect number 2's DNA profile. And this is the DNA profile for suspect number 3, which is unfortunate because it matches the DNA profile from the crime scene. This gel proves that suspect 3 was definitely at the crime scene. When you look at the gel that was created to determine paternity, the bands of DNA fragments from the child have to match at least one of the parents' bands. So this first band of DNA fragments from the child match male number 1. The second band matches mama. The third and fourth bands match male number 1. The fifth band matches mama again. If you keep going, you can see that male number 1 better be ready to rise above and own the fact that he is now a daddy. All right. Be ready to explain the procedure for creating a DNA profile, and also be ready to practice analyzing DNA profiles to determine paternity and suspects that were at a crime scene. Until next time.